Welcome everybody to our latest Q&A, this time with Professor Simon Carding from the Quadram Institute in Norfolk, I believe.
Norwich.
Norwich. So obviously we're here to chat with Professor Carding about his work. So over to you. If I could pass the ball over to you, perhaps if you could give us an overview of your work there as it relates to ME/CFS.
Okay, so I should probably start at the beginning. So it's probably around the summer of 2014 that Pierre and Richard Simpson from the Invest in ME research charity paid a visit to some senior people at the University of East Anglia, which I'm a member of, with the idea of trying to establish a research centre based on ME at UEA. And I think I was sort of brought in as somebody who might be interested in this because of my interest in immunology, gut immunology, and in particular inflammatory bowel disease, which I've been working on for quite a while. And accompanying me was Professor Tom Wildman, who's a virologist. And UEA was sort of quite lukewarm, fair to be say, I think, about setting anything up formally. However, Tom and myself, after having some discussions, thought and reading a little bit about ME, because prior to that, we'd not come across ME and certainly didn't have any research activity in the area. We decided, well, actually, there may be something worth investigating here. And based on our expertise in viruses, gut immunology, gut microbiology, autoimmune diseases, we thought it would be an interesting hypothesis if we were to investigate the causes of ME being in the GI tract, in the gut, and in particular due to disturbances in the populations of microbes that normally reside in the gut, and changes in these microbes causing changes in the permeability of the gut barrier. And this would cause leaky gut. And this would then allow things that normally don't get into the body to get access to our immune system, which would stimulate the immune system in a chronic manner. And as a consequence of that, we get some of the manifestations of ME. So that was our starting point in 2014. And we then went back to the charity, talked to Pierre and Richard, and we also had conversation with UEA. And we got an agreement for the charity and UEA to co-invest in a PhD studentship. So this was very new to the university and certainly was very new to the charity. But from that small beginning, we now have 3 joint funded PhD students with the university and the charity. The first one of the students graduated this year. We've got manuscripts that are being submitted and under review now, so we'll get some output from that. And we've got other students who are continuing on the track of trying to identify whether or not there are any abnormalities in gut microbes in ME patients. And if the patients as a response to this generate abnormal immune responses that are targeting their gut microbes. And that is analogous to what we believe to be the cause of inflammatory bowel disease. So we're looking at another potential role of gut microbes triggering an autoimmune reaction, but in ME patients that causes not inflammatory bowel disease, but ME.
So from your side of things, do you see inflammatory bowel disease? Well, I think you would see inflammatory bowel disease as being separate and distinct to ME.
Absolutely, yeah. But the interesting thing is, from my reading of the ME literature, is that a majority of patients with ME do have GI symptoms, more commonly irritable bowel syndrome, not inflammatory bowel syndrome. And this seems to precede onset of some symptoms. So there are clearly, I think, a large number of ME patients that have GI abnormalities. And what we understand about irritable bowel syndrome is that there's probably also an active role for gut microbes in triggering or making that disease worse. So again, there's an overlap there in abnormalities of gut microbes causing an adverse response in the patient, which can be immune mediated, causing inflammatory bowel disease, ME. We also believe gut microbes might be actually associated with about 90% of all human diseases. Now, a lot of that is purely association, but for some of the diseases, there does seem to be a solid link between the gut microbes changing their behavior, and that's then related to causing or triggering disease. So that's the sort of pathway or the guide that we have for our ME studies. We're looking for similar evidence from that.
So I guess a big challenge must be to try and tease apart the evidence to find out whether the change in the gut causes the disease or is an outcome of the disease itself, presumably.
Yes, so it's very difficult to dissociate cause from effect, because if you were to think of it in a purely scientific way, you would want to take out what you think the causative agent is from a patient, put that into another individual, see if it causes disease. Now, those experiments are unethical in humans. We can't do that. But you can do that using experimental animals. So you can take what you believe to be the cause of a disease from a patient, put it into an animal. Does it cause the same symptoms? If it's yes, then you've got a causative agent, a potential causative agent. If it doesn't, then that still doesn't rule it out completely. Maybe you haven't got the animal model right or whatever, but you're certainly on track to investigate that. Now, fortunately in ME, we do not have a good animal model of the disease. So it's very difficult to actually do those studies. So what we're left with is trying to tease out what might cause the changes in gut microbes and when that occurs in the timeline for a patient to develop the disease. So the best way we can do that in patients is to do what we call longitudinal studies. So we follow an individual for a period of time, taking lots of samples and really looking for, when does the trigger point occur in disease onset? And is that preceded by distinct changes in their gut micropopulations? So most of our microbiota studies that we do, not just in ME, but for other diseases as well, are based on longitudinal studies, where we can get a clear timeline for some of these events and then trying to link them more strongly with disease onset or disease progression. And the other confounders we have to worry about with the microbiome is we now know that some of the major factors which cause changes in your gut microbes are to do with lifestyle and behavior. So genetics is a small component, but how you live, where you live, really become major determinants that can shape and change your gut microbes. Two good examples: medications, antibiotics, how long, how strong, and infections, GI infections. So you get food poisoning, or you get a virus infection from contaminated food or things you drink. The infections can cause long-term changes in your gut microbes. Medications can have the same effect if you take lots of courses of antibiotics, and the very strong ones. It can take your gut microbiome years to recover from that. Diet is also a modifier of your microbiome, but it's less severe. So your microbes will change quickly to your diet, and it adapts very well to it. But ordinarily, that's a good thing, because your microbes are trying to use what you eat to get their food, but also to process the food to give you the active ingredients from the food in order to keep you healthy. But medications and infections are really bad news for your gut microbiota. So if you get a GI virus infection, rotavirus, for example, norovirus, they can cause serious change in your gut microbes, which can then, if it's not cleared and it persists, can cause all sorts of adverse reactions and effects. Some of those may be related then to ME.
So one of the questions that we've got essentially comes down to, has there been sufficient research done so far to allow you to have an idea of what an average healthy person's gut microbiome looks like? But then, as you've just highlighted, diet and such like can have a quite an effect on that. So it must be hard to be able to say this is a healthy person's, microbiome population.
Yeah, very much so. So an important thing here is that everybody's gut microbes are distinct. It's sort of like a fingerprint. You have a microbial fingerprint, and it's constantly changing in response to how you're living, what you consume, what you eat, what you get exposed to. So it's very difficult from one single sample to determine, well, that's your normal microbiome. It will fluctuate. And we do not yet know what a healthy gut is. We know what a disease gut is. And so all this, a vast majority of studies done so far to try and identify what a healthy gut is based on comparing a disease gut to some healthy, non-affected individual. And then we can say, well, this is a healthy gut and this is a disease gut. But we got no way of knowing that that healthy gut in that person that was analyzed is the same for everyone. It's very difficult. So what it comes down to is to be able to get a clear picture of what a healthy gut is for one person, is to do lots of samples and look at how it fluctuates.
Yeah.
And from that, you can actually find out what the core healthy microbiome is for that individual. So we have to go beyond looking at populations, groups of people, to individuals. And of course, that's very time consuming, it's very expensive, but that's really what will allow to say, well, actually, your healthy gut microbe is made of this, but it's different from the person next to you because they've got a different microbiome. So we've got to start looking at this from an individual perspective. And this sort of fits in with the current buzzword of personalized medicine.
Mm-hmm.
You need to give the patient the right drugs in the right amount. You can't just prescribe the same drug to everybody with the same disease. It will not work. And the thing to remember about drugs, very interesting, is that most of the drugs we take are oral medications. So, of course, your gut microbes doesn't distinguish a drug from food. It will process them and metabolize them just as if it were a food. Now, that can be good because some drugs we take are called prodrugs, which need our gut microbes to process to make them active drugs. A good example of that is metronidazole. But there are some drugs which will get metabolized by your gut microbes to make them toxic. And that's bad, obviously. That's a bad consequence. So again, you don't want to give those sort of drugs to people with a particular microbes that will make toxic products from them. Mm. So I think medicine is slowly coming to realize the importance of gut microbes, and that we have to factor in an individual's gut microbes as to whether or not as to which drugs we give them. And there's some interesting studies coming out showing that anti-cancer drugs working in only some people with a certain type of gut microbes, in others there's no effect at all. And it's the same for cancer drugs, it's also the same for anti-HIV medications. How well they work depends on the patient's microbes. So we're moving towards a personalized medicine, and gut microbes, your microbiota, is an integral part of that in determining what sort of disease you have, but also what's the best means of treating that disease for you.
Okay, thank you. As you were saying that, I recalled that I had seen at least a couple of articles about the cancer drugs being, affecting different people differently, and the articles at least speculated it was down to the gut microbiome, individual gut microbiome populations of the patients. But you beat me to that one, so.
Okay. Yeah, they're called checkpoint inhibitors, the particular drugs that we're talking about, the anti-cancer drugs.
Right.
Checkpoint inhibitors. And the drugs actually sort of modulate the immune system. So again, these drugs target the immune system, and hopefully to focus the immune system on the cancer cell and not healthy cells.
So to go back to the gut microbiome in ME patients, it's a question about, as you've noted, that quite a number of ME patients seem to exhibit GI issues, including intolerances. The question basically is, are those potentially subgroups of the same disease, or are we actually looking at different presentations of different diseases?
I think we're looking at different clinical subgroups. So, we define ME based on symptom severity. That's a convenient way, certainly for research studies. Are patients mild, moderate? Now, I've always wondered, well, what demarks somebody as mild symptoms from moderate symptoms? I mean, on a day-to-day basis, they can be moderate, they can be mild, they could be severe. So that's not particularly good. But then severe ones seem to be far more easily categorized. You're in the bed all the time, you're in your housebound. Nobody's going to argue you're not severely affected if you can't get out of bed or can't get out of the house. And so we focused on the severely affected because we thought, well, the disease phenotype, the features would be more consistent and robust because the patients clearly are all similarly affected. And I think some of the research that's coming up in the mild to moderate group of patients, we're finding some consistencies, but there are also ambiguities and differences. And again, when you look at how the patients have been diagnosed, what criteria they've used, I think if you delve into the data and sort of categorize the data according to patient subtypes, symptoms, severity, duration, etc., even male, female, I think it's becoming more apparent that there are clinical subgroups of ME. And so, how many there are, I don't know, but severes can be clearly distinct from mild moderates, and then within that, you may have 3, 4, or even more subtypes. So again, in trying to sort of group all these individuals into one group to look for a cause, you may just lose the real signal in all the noise because you've mixed everybody up with different symptoms, different duration of symptoms, etc. So I think we've got to be much better at segregating patients according to lots of different criteria to get a clearer picture of what the underlying cause is. And of course, this all relates back to diagnosis. I'm aware of patients can take 5 years or more to get a clear diagnosis. Of course, what's been happening in those 5 years? Their symptoms fluctuated. Have they got better? Have they got worse? Is the same? So, we need to go all the way back, I think, to GPs or whoever's making initial diagnosis, and they need to be better informed about, how do we diagnose ME? We're not just sending them off for CBT or GET or whatever. We actually want to know what's actually the pathological cause of the disorder, and not just pass them off to, psychiatrists, psychologists, or physical therapists.
So you mentioned GPs there. I think it would be a good time to bring in the fact that I believe you're looking to engage with at least what you have a kind of a sponsorship for GPs so they can take up one day with yourselves. Is that right? Yes.
So we've been trying to recruit severely affected ME patients in Norfolk, and our hospital, the Norfolk and Norwich University Hospital, does not have a clinical service for ME. So it's sort of out into the primary and community care. And there are 2 major GP practices in Norfolk where a lot of patients get referred to. And one of these is in Great Yarmouth, Lowestoft area. And the clinical commissioning group there, we've been involved in trying to get them to help us identify patients and recruit them into our research projects. And that's proven to be difficult, I think, because again, we're asking a new process to be put in place that didn't exist previously. We need to get somebody who's medically trained to go out with us to see patients, to allow us to get samples. That's just not in existence before. It's physiotherapists who've been visiting patients, not clinically trained people that can take blood samples, for example. So we've had to invent processes with the CCG. And in the course of that, they've now sort of realized they have a research outlet through the Quadram. And so about 6 months ago, they approached us with the idea of this GP fellowship scheme. And this is for a newly recruited GP to work in the practice out there, but to spend 1 day a week for a year at the Quadram getting involved in our research. So this is biomedical research. We're not training them to do CBT or GET. That's just that's outside of my remit. It's to get them an understanding of what biomedical research is. And so when they go back to their GP practice, they have an idea about, explaining to patients what research is being done, new avenues of research, new treatments potentially that are coming along. So they will serve as, if you like, a champion for ME research, and it's biomedical research. So we're currently, I think the CCG are currently advertising for this fellow, and we would hope to have one in place, pending interviews, etc., certainly by the end of the year, if not sooner. And with the clinical trial that we're trying to get started, it's an ideal opportunity to get a GP and a GP practice involved in that, if not just to help us out recruit patients for the trial. So I think that's an important activity because, we are trying through research to get a little bit more awareness of ME and, the sort of condition that it is at the biological level in the front line, the GP practices. So hopefully that will feed into, a better, more quicker, more accurate diagnosis, treatment pathway, hopefully, for ME patients, at least in that GP practice. And if this works, I think the model could be extended to other GP practices and maybe other centres as well across the UK. So I think that's important.
And
I should say before I forget, that this is a 3-partner investment in the GP scheme. So the GP practice, obviously, Quadram and Invest in ME Research are the partners investing. So we're putting funding in to help this happen.
Okay, wonderful. Certainly from my point of view, that's kind of a step further outside of the typical lab-based work that, well, certainly I would stereotypically have of researchers. So, I personally, I would expect, the patient community will certainly be heartened to hear that kind of extra step is being taken, because so many poor experiences are had by patients when they're interacting with GPs.
Yeah.
But we can only blame GPs so far because of they can only know what they're taught, essentially, to a point.
I know, that's a very good question, Randy. So the other thing we're trying to do is to try and make changes in the medical school curriculum. Again, here at UEA, because I teach medical students, Tom Wildman teach medical students. We ask medical students, how much exposure do you get in your medical education to ME? And it's virtually nothing. So Tom and I are through a particular scheme that the medical students have, which is called research learning. So they spend a period of time studying a research project and make presentations. Tom and I are trying to get more ME-focused research there for the students to sort of investigate, present, to increase awareness. We also have medical students that come and do research projects, anywhere from 3 months to a year in the lab. And again, this gives them exposure to ME patients as well, because they come out with us to visit patients. So we're trying to make inroads into the educational deficit as well for ME. But it's very, very difficult because, the medical school curriculum is so prescriptive and prescribed by the General Medical Council. There's no scope or room to sort of expand and give special series of lectures or anything. So Tom and I are trying to do it through their curriculum. When they get exposed to doing research projects, we can bring ME research into it that way.
Brilliant. What you previously touched on before my diversion, you mentioned a clinical trial. I wondered if you could expand a bit on that for us.
Yeah, so I mean, a lot of thing that's driving our research is evidence-based interventions. I mean, that's sort of the buzzwords, if you like. So we want to develop new means of treating ME, but from research evidence. So the research will underpin a new intervention. So again, So from our gut microbes and looking at the abnormal gut microbiota, we've looked at ways of restoring or replacing an abnormal gut microbiota in patients with a new one, with a healthy one. And what we're proposing to do is a feasibility clinical trial to do a fecal microbiota transplant therapy in ME patients. Now, this is not new. There's a Professor Barodi in Sydney who's been doing this for quite a while, who's claims, some quite striking effects of giving patients with ME an FMT, a microbiota transplant. So he's been advising us on developing the trial. He's been very, very helpful. And what he's done is sort of lots of individual patients, no proper trial. But based on that, we thought, well, there's evidence here, and from our own research, to see if we can do a feasibility trial. So that's what we're planning. Invested in ME research, raised a lot of money towards this. So we're still trying to fundraise to get all the money we need to do the trial. We're developing a facility within the Quadram to be able to do FMT, because the MHRA, the Medicines and Health Regulatory Agency, said FMT is to be treated as a medicine, as a drug. So that means it has to be prepared under very stringent conditions in a licensed room by licensed practitioners. So we're investing in a facility to do this in the Quadram. And we hope that the first FMT trial we'll do is in ME patients. So if everything goes well, we get the rest of the money. We're looking to probably start the trial about this time next year.
Okay. Certainly, I'm not sure if you're aware of it, but within the last day or so, there was a report from the States, I believe, about essentially a fatality that came about through FMT. But clearly, the report of that fatality made it clear that essentially the proper or stringent testing wasn't done for, I forget what I think it was a bacteria, antibacterial resistant bacteria was present, and then obviously it was put into somebody with a compromised immune system.
Yep.
Unfortunately, it then played out from that.
Yep.
So while frustrating as it might be to view FMT as a medicine, presumably that is one of the main reasons why, to make it as safe as is possible, given the current technologies and testings available to us.
Absolutely. I mean, as you know, FMT is used in the NHS now in the UK for treating C. difficile infection. It's highly effective, 90% or higher cure rate. We are working with the Norwich Hospital to allow them to do FMT in their patients. We've done about 25 patients so far, but we are very, very aware of potential risks. Fortunately, there's not been any adverse effects from the vast majority of FMTs done in hospitals. I mean, donor selection and criteria is incredibly important. We started off basing the donor criteria on the same criteria used for a blood donor. But then as we learned more about the microbiome, that has extended now into even doing psychological profiling of the donors. We look for, as more and more pathogens are identified in the gut microbiome, we're including those in the screens. So it's very, very rigorous. And because it's going into patients, they have to be assessed as being safe for what we know about pathogens. And contrast that to, home DIY FMT, which is on YouTube and everything. Oh, that's incredibly dangerous because of the risks, as you just said. If you don't go through these very careful and sophisticated screening tests, you don't know how safe the donor is. And the handling and the processing is also done under stringent conditions. So again, doing it on your kitchen tabletop is not something I'd recommend either. So there are reasons why, you know, the regulatory bodies are concerned about having stringent safety precautions and screenings in place. But when you step beyond that and you're looking at private clinics, home, whatever, then, you know, all these risks come to the fore much more.
Okay, wonderful. So if people were interested in taking part in this clinical trial to be, perhaps you, is there a route that they could take to register interest or get some kind of referral or?
Yeah, so we have a long-standing relationship with the clinical fatigue service at St Helier and Epsom Hospital. And until recently, the head of that facility was Dr. Amalak Bansal, who's now sort of moved away from that and is still active, but he's no longer the head of that institute. So the arrangement is still that we will utilize Dr. Bansal as a source of patients. He's still seeing ME patients, St. Anthony's Hospital primarily, but he will identify the patients that are best suited for our clinical trial. And so he has contacts and details and access to a large number of patients from his St Helier service. He has still strong links. He works 1 day a week, I believe, at St Helier. So we would envisage that that would be the route by which we would recruit patients. And so it's either through the CFS facility at St Helier and or Dr. Amalak Bansal. So we at Quadram would not be involved in identifying or selecting patients. That would come from the clinical service using the criteria that they've used for a long time to diagnose and assess ME.
Okay, wonderful. Thank you for explaining that. Pretty much running out of conversation points that I think would be useful at the moment. A couple more quickly. One is that one of our forum members believes that the Quadram were at some point researching a leaky gut blood test. Are they correct in that recollection? And if they are, is there any news on that you're aware of?
So that's half right.
Oh, okay.
I mean, I should say that there is no gold standard for diagnosing leaky gut. It's indirect measures. The NHS doesn't endorse or accept any test at the moment as indicator of leaky gut. So we rely on sort of surrogate markers. And as I've said, I think earlier, that if we can detect microbes or microbial products in the blood, that's usually a pretty good indication of a leaky gut, because most of those microbes we think come from the gut. Now, there are sort of sugar-based solutions that people can use. In America, they have a lactulose mannitol test, which is a drink that people take, and then the urine sample. If one or more of those sugars is in the urine, then that's an indication of leakiness of the gut. So, but there's no gold standard for that. So what I think the question refers to is looking for evidence of the exposure of the immune system to gut microbes and their products. And what we're using as an indicator of that are the presence of antibodies, which would be in the blood, that react with microbes in the gut. So one of the projects that one of my PhD students is undertaking, looking at whether or not a patient's blood antibodies react with microbes in their stool sample. Now, ordinarily, you would not expect your immune system to react with your gut microbes. So we're trying to ask the question, are there gut microbe-reactive antibodies in patients? If the answer is yes, then what are those antibodies reacting to? Can we identify the microbe? Because if we can identify the microbe, then that could be a strong indicator of that microbe causing maybe some of the symptoms. Of course, if we know what the microbe is, we can target that microbe to get rid of it, kill it, whatever ways and various means, antibiotics being one, for example. So if we can identify what causes the immune response, we've got clear insights into stopping the immune response. and hopefully treating it. So I think that's what the question refers to. So it's an antibody detection system to determine whether or not they're reacting to gut microbes.
Okay, wonderful, thank you. Another one which has become a bit of a interest of patients at the moment is the theory that there is something in the blood affecting mitochondrial function.
Right.
Now, a number of groups have found evidence that points towards this, and there's the suggestion that microvesicles may be involved, which I believe that one of your papers looked at.
Right. Yep.
Now, obviously, that's moving quite a bit away from probably your focus of your research. But could you see that something produced in the gut that is then released into the bloodstream through a leaky gut could then be having an impact in such a way? I mean, just to briefly explain this, something in the blood, patient's plasma has been removed and replaced with healthy control plasma, and the mitochondrial functioning in the cells that this plasma is then surrounding the cell, the mitochondrial function seems to return. And if you do the reverse, then the mitochondrial function again is impaired, or if it's healthy control cell, then patient plasma will impair the function of those cells, mitochondria, or the function of the mitochondria. So obviously we don't know what the something is.
Yep.
So that really is, in regards to that, that's the big question. And could microvesicles being released by intestinal microbiota, could that be a route potentially? Obviously, it's full of ifs and buts, but
yeah, sure. No, that's a really interesting question. So we are interested in bacterial-derived microvesicles. We're about to start a preliminary study using some samples we've obtained from patients to look for the presence of bacterial microvesicles in their serum, in their blood, as an indicator of leaky gut, but also maybe a source of stimulating the immune system. And this might even prove to be useful as a diagnostic tool in ME. We don't know. So that's something we're just about to start. But I think it's worth noting that microbes are not directly in contact with your gut cells. So you have this barrier in your gut, which is made of epithelial cells. But on top of that, you've got a sticky gelatinous material called mucus. And bacterial cells cannot get through the mucus. So they sit at the surface. So you have like a sterile layer of mucus that separates your epithelial boundary cells from your gut microbes. Now, so then the question is, well, how do gut microbes communicate with cells of the body? How do they deliver signals? And we think there are two ways. One is through metabolites, so small compounds that they secrete, metabolites that can get through the mucus into epithelial cells and maybe the bloodstream. The second route is through microvesicles. So these are nanometer, very, very small spherical particles that bud off from the surface of certain types of bacteria. And we've shown they can permeate through the mucus into the epithelial cells and actually can stimulate the immune system. Now, we believe that these microvesicles play an important role in instructing the immune system to not react to gut microbes and food. Because you imagine if we had an immune reaction to everything we ate and to all our gut microbes, we'd be in a hell of a state. We'd be a mess. So we believe the microvesicles are important to instruct the immune system to ignore gut microbes, food particles. And so they make the immune cells produce what we call tolerance. So tolerant to everything in the gut. Now, what we've seen in inflammatory bowel disease patients is that the immune cells lose that ability to respond to the OMBs to be tolerant. Instead, they now become super inflammatory. immune cells. So there's a breakdown there in the communication of these microvesicles with the immune system in IBD. We're looking for similar evidence in ME patients. So is the immune system in ME patients now abnormally responding to the microvesicles, particularly those that you've seen in the blood? So microvesicles could be important. There's another source of microvesicles that our own cells in the body produce. These are called exosomes, which are very popular in cancer research at the moment. So these are vesicles that all cells in the body, our body, produce, and a lot of them end up in blood, saliva, breast milk, sperm, any lymphatic fluid. Any fluids in the body will contain these exosomes. And we can distinguish exosomes because they're unique to the cell that produces them. So there is, if you like, they're a signature of the cell that produces them. And we know in cancer that cancer cells produce these exosomes, and the exosomes promote the growth of the tumor cell, metastases spread around the body. So there is a group in the US that's starting to look at exosomes in the blood of ME patients. Now, are they unique? Are they distinctive? What cells are producing them? You know, and are they attributable to any of the causes of or pathology of ME? So there's a lot of interest at the moment. It's all new research about microvesicles. We're focusing on the bacterial type. There are groups, as I said, in America looking at exosomes. So all of these are present in the blood. So I think it's a very interesting avenue of investigation in ME. And again, other diseases are sort of pointing the way here. Cancer, inflammatory bowel disease. So, you know, so there is a precedent.
Brilliant. Okay, well, thank you for explaining. I think that brings us to the end of what I'd like to cover at this point. So just to finish us off, is there anything about, do you have any planned work that you haven't mentioned yet that you can talk to us about, or do you have any ongoing work that you'd just like to briefly talk to us about?
Yeah, so I hope I haven't given the perception that we work in isolation here in Norwich. We're not interested or collaborate with anybody else. We do, in fact, collaborate with a lot. So we have extensive collaborations with Sweden, Norway, Denmark, other places in the UK, the USA. And we're currently involved in an FMT trial, which is currently ongoing in ME patients in Norway at the moment. So we are working with them on analysing what's happening in the gut microbiome of patients after they receive an FMT. So we're just about to get our first set of samples. So that's very exciting. And we're also collecting stool samples from ME patients in Denmark, Iceland, and Norway. So we are sort of becoming a stool repository, if you like. But you know, it's all for the benefit of our research, and also, you know, it shows that there are other countries and other ME research groups elsewhere in Europe, particularly, that are very interested in working with us on this. So it is the sort of multinational research activity that we have, and Norwich at the moment is sort of the hub of that. But hopefully, you know, we'll be able to develop ME research in other countries as well through interacting with them.
Brilliant. Fantastic, thank you. So finally, if people want to support your work, or patients that is, through donations, where should we be directing them to invest in ME? And
invest in ME research.
Yeah.
Invest in ME research. So we're working with them to try and raise the additional money we need for the FMT trial. So donations will be very, very gratefully accepted by the charity. And as I say, virtually all the money they raise goes into research.
Right, wonderful. Well, what we'll do in the video description, so anybody who wants to support, we'll put a link there through to the Invest in ME donation page, which I'm sure they'll have.
Absolutely.
And hopefully we'll see some contributions there.
I hope so, Andy. I hope so.
Right, thank you very much for your time.
Oh, you're welcome.
Wonderful.
Okay.
Thank you.
Thank you.